Okay, today we'll discuss about yeast production and different type of yeast we get through this particular production process. So in last class we have mentioned about the type of two types of yeast generally we grow, and Saccharomyces cerevisiae and Candida utilis. Saccharomyces cerevisiae mostly we use for food supplement or generation of different nutrients, whereas Candida utilis or Candida is actually used for animal food production. So the production process will require substantial amount of carbon, and carbon generally provided by using molasses. So when you use molasses, you will get substantial amount of carbon, but you still need phosphorus and nitrogen. So for supplying phosphorus and nitrogen, we use orthophosphoric acid or ammonium salt of orthophosphoric acid. And this process of production depends on multiple stages. So first we prepare the inoculum. So inoculum preparation is done in the laboratory scale. So you get your slant grown on test tube, and this fungal slant. From this slant, you will just take one lookful fungal strain, and then you grow that in a conical flask in laboratory. So first stage is inoculum preparation. So inoculum preparation is done in a smaller scale, and there you can use nutrient media to just grow them in a small vessel. And then after growing the inoculum in the small vessel, to transfer it to a larger tube. These are actually part of upstream process, and from F2 you transfer it to another fermenter. Then you have a little bit bigger fermenter. And this way you can have one more fermenter, that is F5, and finally you'll have the industry scale fermenter. So all these fermenters and in the final fermenter we'll also have some other line. spent molasses will go to waste treatment plant. Okay, so in this particular scheme, first we grow the inoculum in the laboratory condition, and then we transfer it to a little bit bigger fermenter, because we always try to get 10% of reactor volume as the inoculum size. So this F2, F3, F4, F5, they're all actually having uh, different sizes, and ultimately the entire volume will go to the next stage as inoculum. So finally, the inoculum will get in the final stage at F5 will be transferred to F6. Now one can ask that the entire operation could have been done in a bigger reactor, like if you could merge F2, F3, F4, F5, then also you could generate the inoculum. So, but that generally we don't do, because whenever you try to grow them in a larger vessel, you will not be able to maintain appropriate environmental condition required for their growth. And while preparing them, it is always better to start with smaller vessel, and where variability will be less. And also, while preparing them, you mostly run them in fed batch mode. And so you try to monitor the nutrient requirement, and you keep on adding intermittently certain amount of nutrients to the system. And at the end, when you run the final reactor system, there you have option to choose whether you will go for fed batch or continuous mode. That depends on the kind of final product you require. So in some cases, you may not require really very uh, purified product, and this production process is really monitored in a sophisticated manner, where you will ensure very small amount of residual carbon or residual substrate. Then you can go with continuous operation. Otherwise, you can keep on operating this reactor also in fed batch mode. So while operating all this inoculum preparation reactor as well as final fermenter, few control parameters are continuously monitored. One is pH, then temperature, antifoam. Antifoam is more important for the final fermenter, and also the substrate concentration. Substrate concentration, mostly sugar concentration, you try to monitor, and then make sure that you have very small amount of residual sugar. And target residual sugar for all fermenter should be less than 0.1%. So that will be your residual sugar in the fermenter. So you maintain the concentration in such a way that the residual sugar in the reactor would be less than 0.1%. And in different tanks, when you try to just generate the inoculum, there you just use corn strip liquor, or sometimes you also use little bit of mixture of molasses like beetroot molasses and cane molasses, and then you ferment it. So in the first 5 stage, you have intention to grow same species at a higher concentration, and the final fermenter, you maintain your environment in such a way that you get substantial amount of biomass, and also very good reduction of sugar or very good utilization of sugar. So when you have this very good utilization of sugar, essentially most of the sugar will get converted to biomass. And while doing so, you have to make sure sufficient amount of oxygen is also maintained in the reactor system. So typically in larger fermenter, we try to maintain 1 ppm. So 1 volume of air per 1 volume of media or broth per minute. So 1 ppm is your typical air supply rate. This will actually ensure that the system or the biomass present in the system should get at least 0.2 molar or 0.2 millimoles of oxygen per liter per minute. So you maintain air flow rate in such a way that the oxygen utilization rate or oxygen uptake rate would be 0.2 millimole per millimole of oxygen per liter per minute. And when you talk about nutrients, the pH generally in all reactors we try to maintain in the range of 4 to 6. That is the optimal pH range we try to maintain in the reactor. And this sugar monitoring is done by you can use electrochemical probe or you can use offline monitoring, like every half an hour you will just collect sample and do the sugar analysis to estimate sugar concentration. And typically in this fermenter system, you have option to choose molasses obtained from beetroot molasses, like from in different sugar making process, people can use beetroot or cane sugar, cane for making sugars. Now, when you use beetroot, there you will end up by getting beetroot molasses. When you use sugar cane, then you will get cane molasses. And both of them are quite important. So if you really need to get good quality of yeast, then it is better to use a mixture of beetroot and cane, beet molasses and cane molasses, because this beet molasses has almost 5 times higher nitrogen content than cane molasses. So in content, in beet molasses is almost 5 times higher than cane molasses. Whereas vitamin content and other trace nutrient content like biotin is more with cane molasses. So trace nutrient are more in cane molasses. So nitrogen content is more in beetroot molasses, whereas trace nutrients are more in cane molasses. And sugar content wise, cane molasses is more preferred. And also cane molasses has many other trace elements like magnesium, calcium, some other vitamins. So biotin is one of the prominent candidates, but apart from biotin, you can get some more vitamins and trace element in cane molasses. So when you talk about ratio, at least 20% of cane molasses is preferred to provide sufficient amount of trace nutrients. So in a given mixture, when you try to grow the yeast in final fermenter, there you get the cane molasses. So you use a clarifier to clear the purify the cane molasses and beet molasses. Then you have a sterilizer to sterilize them. Then you have a mixing tank, which is also known as wort tank, WORT, wort tank. And then in the mixing tank, you mix them together and finally you transfer them to the final reactor. And while transferring things to final reactor, externally, you add, because it is the sufficient amount of nitrogen may not be available with the molasses, so you need to add some nitrogen in form of ammonia or ammonium ion, and also you add some phosphate, again some phosphate salt or orthophosphoric acid or ammonium salt of orthophosphoric acid, and if required you can add some other trace nutrients like other vitamin and trace nutrients, but requirement is less if you just maintain 20% minimum 20% cane molasses as your main carbon source. And in the large fermenter, as we are supplying air supply will ensure good mixing and also to provide oxygen, and you may have stirrer also inside the reactor. So whenever you have a mixing using stirrer as well as aeration, due to abrasion, you will have cell disruption. And naturally, cell will also die after attaining certain age. So when you have cell disruption, proteinaceous substance present in the cell will come out and they will actually get mixed in the broth. And whenever you have hydrophobic hydrophilic interaction, and due to presence of this air at the top of the liquid layer in the fermenter, you will have foam formation. So foam formation is not desired in any other fermenter, because wherever you have foam formation, oxygen transfer from the dead space gas will get hindered, and also this foam will try to collect biomass present in the system. So whatever biomass will go to the top surface of this liquid, they'll automatically get trapped in the foam. So foam formation is not very desired, and also effective exit of carbon dioxide will also get affected if you have foam formation on the top. So to avoid getting foam formation, one option is to have a sensor which will sense the level of broth. So you have a level sensor at the top, which will actually send a signal to the controller that broth level has already reached to a specific height, and then it will activate a pump which will add certain amount of anti-foaming agent. So this anti-foaming agent will try to reduce the surface tension, and it will dissolve this foam, and ultimately the foam on the top layer will get cleared. Only problem with anti-foaming agent is these are all inert substance, but they will also get mixed with the broth. So silicon oil is one of such anti-foaming agent, but only problem is in downstream operation, if you really need to get pure substance or pure biomass from this anti-foaming agent will create little bit of problem while you do the downstreaming operation. So to avoid using anti-foaming agent, you have another option to use a foam disruptor. So you will have a mechanical foam disruptor. That means on the motor shaft, you will have one, means horizontally one needle will be mounted on the motor shaft, long needle. So whenever it will rotate, it will actually puncture the bubbles or foams. So mechanical foam disruptors are not as efficient as this anti-foaming agent, because anti-foaming agents are chemicals which reduces the surface tension, and they can do it very quickly, whereas mechanical disruptors will take longer time to disrupt the foam. But with that, major advantage mechanical foam disruptor is that you are not using any extra chemicals. So ultimately in downstream operation, you will not have much problem to deal with the solution, because you are not having any external agent present in the final solution. So after getting the biomass grown in this system, you get in the broth, you get around 5% concentration. The solid content of the broth would be around 5%. And after getting that 5% biomass concentration, you send it to a rotary vacuum dryer, because you need to just concentrate the biomass. Your ultimate objective is to get solid fraction, not the liquid. So after getting the biomass, if you run the system, say if you're running it in a fed batch system mode also, after 10 to 20 hours, depending on the strain you are using, 20 hours of operation, you can get 5% is concentration in the broth. Then you send it to the rotary vacuum dryer. So rotary vacuum dryer is one of the downstream, one of the separation device which is used heavily in many industries, and it looks like this. If you take the side view, then you'll see this kind of view. So you have a tank. So front view will have this kind of image. It is very similar to rotating biological contactor. Only difference is this entire inner cylinder is having a porous screen on the outer surface, and in the porous screen also you will have some membrane cloth wrapped on the surface. So the entire cylinder will be wrapped with a membrane surface, a membrane, porous membrane, and both the side will be closed, and here you will have a suction line to maintain vacuum inside. So you'll have two chambers, and in the second chamber you will have a vacuum pump connected. So vacuum pump will create vacuum in the first chamber, subsequently second chamber, and then same vacuum condition will be maintained in this entire drum. So in inner, in this inside the cylinder, you are creating a vacuum, so it will always try to get liquid through the membrane surface. So whenever liquid will pass through, solid will actually get accumulated on the surface, and this drum will keep on rotating. So all the surface will get exposed to this liquid. So this liquid slurry has solid liquid mixture, and whenever you rotate the drum, all the surface will get exposed to this liquid solid mixture, and because of this suction pressure, liquid will move through, because the pore size of the membrane filter is such a way that it will not allow the solid to go in, but it will just take the liquid, and liquid will be collected at the bottom, and from bottom also you have another line to drain the liquid. So there also you'll have another pump to suck the liquid from the cylinder. So you can operate this particular rotating vacuum dryer or vacuum filter. It is called a rotating vacuum filter or dryer. So the upper surface will get covered by the solid. So to remove the solid, you will have some other mechanism. So if I show you the side view, then you can actually see the details. So if you rotate in this direction, the entire drum will rotate. So initially, here you have clear surface, it will enter the liquid, and then this filtration will start. Liquid will go in, but solid cake will deposit, and slowly this, when it will rotate, solid cake, the disc cylinder having solid cake layer on the top will come out of the liquid. Here we will first put some jet spray to dislodge the residual liquid present in the cake. So here this spray will have clear liquid, this liquid only, but clear liquid obtained from the inside of the vessel through this pump. So essentially this liquid spray will ensure dislodgement of all residual liquid from the system, and then you will have another wash cycle introduced by clear water, and then finally you will have a mechanical scrapping device like a small blade. So depth of this blade can be varied, so you will make sure that it will not touch the filter cloth, but you will maintain minute gap between the filter surface and the blade surface. So whatever residual cakes are there, they'll automatically get removed by this blade. And just after the blade, you will have a collecting tray. So whatever you are removing, that will not go back to the liquid. It will get collected on a tray, and then you will just take the cake and process it subsequently. So advantage with this particular system is it's a continuous system, because continuously surface will get renewed, means cake will get removed, so you'll get new surface, and that new surface will again expose to the liquid slurry, and fresh cake will form, and that cake will again get removed by the blade. So when one particular cycle, this drum will complete, by then the initial surface area will automatically get renewed. So instead of operating in a batch mode, you can actually run it in a continuous mode to filter the liquid and separate the solid. So this is one kind of mechanism to actually get a continuous operation of solid separation. Only thing is this cake will not be 100% moisture free. You will get substantial moisture here in this cake. At least more than 10% moisture will be there in this cake. Now you also have another option. Instead of rotating vacuum filter, you can use centrifuge. Centrifuge means you will have continuous centrifuge. Different type of centrifuges are available. Basket type centrifuge is a simple type of design where again you have a cylindrical basket inside a tank, and this basket will be rotated at a particular speed, and you will introduce the liquid inside the basket and whenever it will rotate, because of centrifugal force, the liquid will pass through the filter. They will actually go to this inner wall, and then liquid will pass through the membrane. Solid cake will get deposited at the wall, inner wall, and after a certain period of time, you just stop the centrifuge, remove all the cake, and again start the operation. So this kind of basket type centrifuge is not a continuous centrifuge. You can have disc stack centrifuge where you can operate continuously. So you can have option to use continuous centrifuge or batch centrifuge, or you can use plate and frame filters. In industry, mostly they prefer to use plate and frame filter because operational cost is very less. Only problem is it is not a continuous process. It is again a batch process. So here you will have two frames. In between frame you will have a filter cloth. whenever liquid will introduced, the filter cloth will help to separate the so if you just see two different frames, in between two frames you have place to space to keep the filter media. So if I draw it separately, you have two frames. In between frames you have a filter, and both the side you have filter is kept at the middle of these two plates, two frames, and both the side you have some space. So in one side you will just introduce liquid slurry where you have solid and liquid mixture, and other side you will actually get filtrate collected on the other side. So you keep on adding your slurry by maintaining certain pressure, and following dead end filtration, it will get filtered through this filter paper, and ultimately cake will form at the one side of the filter, and you can keep on running the system until you have complete formation of cake, or that entire space will be occupied by the cake. So when the entire space get occupied by the cake, then you stop the operation, dislodge these frames. That means you just dismantle the frame, then try to remove the cake from one side, and then again you reassemble to get the operation started again. So again, this process is also a batch process. It's called plate and frame filter press. This is also a batch process, but it is very simple in operation, and also it is very economical in operation. Only difficulty is it is batch process, so productivity will not be as good as continuous process. So the rotating vacuum filter will have more productivity compared to this plate and frame filter. But here the operational cost as well as maintenance will be significant, and you need to maintain vacuums. Obviously, it will be more energy intensive, whereas plate and frame filter is not that energy intensive. Only problem is every now and then you have to just dismantle and reassemble. So after getting the broth from this reactor, you actually have option to use different kind of filter, plate and frame, or rotating vacuum filter, or centrifuge, continuous centrifuge. So continuous centrifuge is also expensive because you have to employ certain amount of energy, whereas plate and film filter is the least expensive method. And wherever you separate biomass, these biomasses are actually very compressible in nature. So if you get really very sticky cake on the surface, it will be very difficult to remove them. So deliberately, wherever you are not interested to collect the biomass, there you can just add some other solid material called filter aid. So filter aid are some solid material which will essentially give little bit of mechanical strength and keep some pores in the inside the cake. So suppose you just imagine you are trying to separate some algae which are very slimy in nature, and they're also very much compressible. Means if you just create a lot of compressive force, if you just apply a lot of compressive force, it will get squeezed and they will make really very compressed cake. And wherever you have really very compressed cake, penetrating that cake will be very difficult. So very quickly you will have clogging of the filter pore, and again you have to clean the filter surface to get filtration continue to continue the filtration. Now if you use filter aid, it will actually create some pores in between. So imagine you are just putting some glass beads along with your slimy algal biofilm. So the glass beads will stay in between the biofilm because you will not be able to crush the glass beads. So whenever you have this kind of arrangement, you can always get some channel created in between the glass beads. Filter 8 will actually do similar kind of job. It will create some pores or channels in between the compressed solid layer, and it will make the filter cake permeable to the liquid. So you can operate the system longer period of time. But only thing is you have to make sure that you're not using that filter cake, because filter 8 is also another contaminant, and you don't want to have filter 8 in the system. So this filter 8 is only used wherever you are not willing to use the solid media. You are just interested to use filtrate. Whereas in for yeast extract or yeast production, we don't use any filter 8. We just try to use any of this filter system and then recover the cake, because then we process the cake in different other systems for harvesting the yeast. So I mentioned in the last class also, when you try to harvest the yeast, your intention is to just remove the moisture. So this process of fermentation is also exothermic process. Whenever yeast will form, certain amount of heat will also generate. Now, to reduce the temperature of the fermenter, you need to provide coolant also. So the final fermenter will also have thermal jacket. Through that thermal jacket, you can supply coolant to maintain the temperature in the reactor. And that is true with all other seed fermenter also, but only thing is seed fermenter are smaller in size, so the temperature maintenance is much easier. Whereas in large scale fermenter, you have to ensure that coolant is actually, or coolant coil in placed in the reactor in such a way that it is doing the heat transfer very efficiently. Now, after filtration, filtration is actually, you can say, concentration process, means you are trying to concentrate the biomass. And then while concentrating it, you are just trying to wash it also. So either you can have two different filter system, and in between two filter system, you will have one wash cycle, like washing of solids by clear water. So just to remove the residual molasses, because this after washing, I think I have shown here also one stream. One stream is actually going to. So after washing and concentrating it, one stream will go to waste treatment plant. So whatever wash liquid you will get, that will be your spent molasses. So residual broth and other nutrients will be there in that stream, and it will go to the waste treatment plant for further treatment. And cake will actually have further processing to remove moisture to get different kind of material. So when you use compressed yeast, we are taking this concentrated yeast, we use a cooler to reduce the temperature, and after cooler, whatever yeast we will get, that can go to different kind of, that can go through different kind of unit operation to get different type of yeast. So when you try to get compressed yeast, that means you just have some dewatering, and you'll still have substantial amount of moisture. So final product should be maintained in a refrigerator. Otherwise, that yeast will get spoiled. So you will do dewatering. after dewatering, we'll put them in a mixer to just crush the cake to get uniform size. Now, after getting this mixed, because there actually will get uniform distribution of moisture in the cake. As it has substantial amount of moisture, you can push it through specific extruder, so you'll get different size and shape of yeast. You can get yeast strand, you can get yeast pellet. So you put a specific extruder, and then you go for packing them. And this one you have to keep at 4 degrees centigrade. You have to keep it in a refrigerated storage system. Then same thing, you will have dewatering. Because dewatering is again having through centrifuge or filter press. So in all process, you will have dewatering when you try to get active dry yeast. Active dry yeast when you try to get, then you get dewatering, and then you have same extruder. Then you have one more drying operation, and then the final dried material will be sending for packaging. And this one you can maintain at ambient condition. Because moisture content is less, so you need not to spend too much of effort to uh, prevent spoilage of this. And then finally you have nutritional dry yeast. Mostly we
Sir,
use.
but is this dry is will be living, or they are dead cells?
So they're live cell, because you are not removing the moisture entirely. Because if you remove the moisture completely at normal condition by maintaining high temperature, then obviously you will be killing them. Our intention is not to kill them. So next class I'll actually mention the use of all this type of yeast. Why we call them as active yeast, why we call them as compressed yeast, why we call them as nutritional dry yeast. This we'll discuss that in the next class. Their importance, and also why do you need to prepare different kind of yeast product in the same manufacturing process. We'll discuss that in the next class. So nutritional,
Okay.
nutritional dry yeast is actually having very minimal moisture content, because if you can remember in the last class we also mentioned yeast extract. So nutritional dry yeast is actually essentially to produce yeast extract. So here you can kill the system, you kill the organism, but obviously you have to make sure that killing is not happening at very high temperature, because high temperature will also damage the other nutritional elements. So if you talk about different vitamins and other biomolecules, they'll also get damaged if you raise the temperature at a very high level. So essentially, we just increase the temperature above 80 degree. So it is one kind of pasteurization only. So it is a pasteurizer. And after pasteurization, we again use little bit different. We will discuss that also, drum dryer, where you can actually supply little bit of heat also. And then you have grinder, because we are trying to get extract. So we try to get the powder from grinder, and then we send it for packaging. And this can actually, because here we'll have very minimal moisture content. So if you have very dry powder, it will try to absorb moisture. So when you try to store it, you have to make sure that you are doing it in a dry storage condition. Otherwise, it will attract moisture, and then other bacteria and other contaminant can actually spoil this material. So moisture content wise, the compressed yeast has maximum moisture. Then little bit lower content is there in active dry yeast, and nutritional dry yeast is actually have will have very minimal moisture content. In the next class, we'll just discuss about different type of yeast, and also why do we need to prepare different type of yeast, and what kind of use we have for different type of yeast. Anybody has any question on this?
In pasteurization, all the bacteria, like we do pasteurization of milk, so in that process, all bacteria are killed. Uh, but as you mentioned here, that yeasts will not be killed.
No, no.
So
Here for nutritional dry yeast, we are trying to kill the yeast. Because essentially, will take extract of yeast. So you will do the disruption.
okay,
And pasteurization is done because when you talk about nutritional dry yeast, essentially your end product is actually some dry powder which will either add directly to the food, or you'll have it as a food supplement. So you have to make sure that no other pathogens or contaminants are actually present in this material. So that's why you do the pasteurization to ensure that you have eliminated the possibility of having any other contaminants, and then you break the cell wall, like you do drying, then you do crushing, grinding, then finally you use the extract powder. So essentially, in the last stage, you are actually in the for this one nutritional dry yeast, you are not dealing with the live yeast cell. You will use yeast extract.
okay, sir.
Sir, what is the long-term effect of moisture in yeast? Means why it is important to remove the moisture?
So if you suppose for any substance, even if you just consider bread, forget about yeast now, just consider bread. You try to dry the bread, and it is common in different parts of India where people just try to sun dry the bread, and then they use it as a different kind of food, like papadum. Papadum is one kind of dried material. Now, in when you keep anything in dry form, longevity of that particular material will increase, because for growing bacteria or fungi or any other biomass, they will require certain amount of moisture, because water activity if not present, then cell will not be able to sustain. So on any dried substance, you will not be able to grow any other organism. And wherever you have growth of other organism on a bread, that means your substrate or bread will get spoiled. Same thing is true with this yeast extract or yeast material. So if you have substantial amount of moisture present in yeast, I mentioned in the earlier class also that yeast is a good source of carbon. It will also provide good amount of trace nutrients. So it is actually essentially you're creating a good environment for creating or growing other bacteria which will actually eat the yeast, or which will actually live on yeast, and then it will contaminate the yeast. So to avoid getting any contamination, we try to remove the moisture and keep different kind of dried condition. Like here, where you have higher moisture, you maintain low temperature to ensure that no other organism will grow at that temperature. Where you have reasonable amount of dry condition, there you maintain ambient condition, because you know that that much moisture will not allow any other organism to grow. So to avoid spoilage of yeast, you need to remove moisture content based on your storing conditions.
Okay, sir.
So, in the final fermenter, what is the impact of the aberration of the cells?
So in the final fermenter, we don't have significant vigorous mixing created by the agitator or aeration line. Reason is our intention is not to kill the organism. So we have idea about the shear sensitivity of the organism. So we maintain this condition in such a way that combination of aeration and agitation will not create any cell disruption. But if you have shear force here, like if mechanically, if you just think you are having a rotating system inside the reactor, so whatever abrasion cell biomass will experience where they are very close to the stirrer, definitely they will not experience similar kind of cell abrasion if they will just go away from the stirrer. So some organism may experience that, and that level may not that abrasion stress or the shear stress may exceed their tolerance limit, and in such cases you will have disruption of cell, but that amount will be very small. And another reason for having cell disruption is cell age. So whenever cell will attain its full age, then it will have its natural death. So whenever it will die, automatically cell will get disrupted, and then inner material will come to the liquid. That's why we will have the formation of foam because of presence of different proteins. So it is not intended to have really very good abrasion of cells to have cell disruption, but it is inevitable because when you run the system, automatically few cells will die and few cells will actually experience some higher abrasion, and automatically the cell will get disrupted, but majority of cell will stay intact. So when you talk about percentage, we get 99% cell as intact biomass, maybe 1% or less than 1% will get disrupted.
Got it, sir.
Okay. Okay. Any other questions? Okay, then we'll meet again on Tuesday. Have a nice weekend.
Thank you, sir.
Thank you, sir.
Okay, thank you.
Thank you, sir.